All right, so today in lecture we're going to look at the rise of land plants. We got into them a little bit last time with the bryophytes, and I'll wrap that up shortly here. And we'll move on to the plants that are most conspicuous in the terrestrial environment and by far the most ecologically and economically important plants on Earth, the vascular plants. And we'll start out first looking at vascular plants that retain some characteristics of the earliest land plants and a life cycle of a fern along those lines. Then we'll introduce a new topic, heterospory, which is really important for understanding the origin of the seed, which is a critical innovation in the land plants. And then we'll start talking about seed plants, and the whole rest of the semester is going to be focused on seed plants. So this is going to be the entry to the rest of the course here. The only group of seed plants we'll get around to talking about today are the gymnosperms, which are some of the more spectacular seed plants, and we'll look at a pine life cycle if time permits. And you'll see that the innovation of the seed leads to some really interesting changes in this alternation of generations. So again, the alternation of generations is going to be really critical to understanding the biology of these plants going forward. All right, so last time I mentioned peat bogs. So this type of moss, sphagnum, that's arguably the most economically important bryophyte. Remember, the bryophytes are the mosses, liverworts, and hornworts. And these bogs are interesting to ecologists and paleobiologists because they preserve a lot of fossil remains or subfossilized remains. Some of these bogs can be thousands of years old and are really important for understanding post-glacial life and can be reconstructed pretty well because preservation in these bogs with their high acidity and high phenolic content from the secondary compounds of these mosses and low oxygen levels really allows for phenomenal preservation. And these so-called bog people I just mentioned right at the end of the last lecture, over 1,000 of these individuals have been found dating back to the Iron Age in northern Eurasia. And as you can see from the person here on the right, the preservation is really amazing. They're pretty well tanned in terms of the secondary compounds. These phenolics basically dye their skin, but in many cases they're perfectly preserved. The bones are a little bit dissolved, but their stomach contents are still evident, et cetera. And you can see the rope around the neck of this individual here. These individuals usually do appear to have been executed, and it's unclear whether they were ritual sacrifices, for example, in some religious ceremony, or whether they were criminals or were basically murdered, or all of the above. But lots been learned about early Eurasians from these bog people. All right, and then I just want to mention briefly that the historical use of peat moss was during the World War I, when trench warfare made wounds oftentimes fatal, even minor wounds, because of the incredibly unsanitary conditions down in these trenches. And sphagnum, for the same reason, as I just mentioned, its great abilities to preserve materials is also has a great use in bandaging. So the antimicrobial properties of sphagnum were put to good use during World War I. And here you see a warehouse full of sphagnum moss that was about to be shipped off to the front, where it was extensively used. So a lot of lives were saved due to peat moss back in World War I. All right, so that's the last thing I'm going to have to say about bryophytes in lecture. And we're going to move on to the vascular plants. And these are the plants that are named for their conducting tissues. Just as we talk about vascularization in animals, we're talking about plants that can move substances around in their bodies readily because they have specialized tissue that allows for that. And the earliest vascular plants go back to a time shortly before these coal or swamp forests that I mentioned briefly, and we'll see a bit more of in a moment. These swamp forests prior to the swamp forest, we see some plants that look like this that are only about often the size of a matchstick, pretty insignificant looking things, but they do have conducting tissue inside their bodies. They don't have any leaves. They don't have, these might look like leaves. This is a little fanciful, actually. They don't have roots either. They do have underground parts, but these are stems, rhizomes. And their sporangia or their gametangia are born at the apices, at the tips of these branches, these swollen areas you see here. And so both gametophytes and sporophytes have been found. And really about the only way to tell them apart is whether they have gametangia or sporangia at the tips of the branches. So these are, they're little tiny plants. They provide us with essentially a fossil link between the bryophytes and the vascular plants in terms of the degree of elaboration of the sporophyte. So remember, in bryophytes, the sporophyte is parasitic on the gametophyte. So the diploid organism is attached to the haploid organism, and it's actually parasitic on it. In the vascular plants, as we'll see, the sporophyte is dominant. The diploid organism is a dominant large organism. But in this particular group of plants, many of them classified as rhiniophytes because they were found in Rhynie Chert in the UK. So that's the part of the world where these fossils were found, just incidentally. But this is there seems to be codominance of the gametophyte and sporophyte generations. So we have something like you saw in the sea lettuce, ova, where you can't really tell the sporophyte from the gametophyte, except that one produces spores, as the sporophyte does, and the gametophyte produces gametes. So this is a brief intermediary stage. These are all extinct. And if we look today at vascular plants, I should mention that these plants, I did say they have vascular tissue, but it's not quite the same as modern vascular plant tissue. So vascular plant innovations are characteristics that we think are responsible in part for their success on land and their dominance on land. It has to do with the dominant sporophyte generation. And this allowed for complex gene expression, diploid organism, 2 sets of each gene. And we also have a well-developed cuticle here. I mentioned a cuticle, this waxy covering on the outside of land plants, is something we see in all the land plants, but it's particularly well-developed in, it's pretty inconsequential in some of the bryophytes, but it's pretty well-developed in all of the vascular plants, as are stomata that I also mentioned, that aren't mentioned here, those pores that have to be present in order for gas exchange to occur when you have a thick cuticle. Vascular tissue, though, is the main diagnostic feature of vascular plants. They're well named. This vascular tissue comes in 2 forms. There's both xylem, which is conducting tissue that's responsible for conducting water and inorganic nutrients for example, from the soil, and phloem, which is responsible for the conducting of sugars and organic substances within the plant. So those are separate vascular tissues, both found adjacent to one another in vascular plants. And there are some mosses, which are bryophytes, that have vascular tissue. And there, these things are called leptoids and hydroids rather than xylem and phloem. But the homology is not obvious between the conducting tissue, or with the conducting tissue in vascular plants. And the conducting tissue of bryophytes is not lignified. So this compound lignin, this complex phenolic compound that is present in vascular plants in the ciliary conducting elements, so in these elements called tracheids that conduct water and inorganic nutrients, is not found in bryophytes at all. And this lignin imparts structural strength to the xylem, and it also imparts decay resistance as well. So I mentioned that in the context of fungi earlier, that fungi, some of them are wood decaying fungi that can break down lignin, and that's really unusual among decomposers in the ecosystem to be able to do that. So the evolution of lignin allowed for a strengthening of the conducting tissues, and we also have the evolution of conducting tissues. This lignin is not present in organisms like this, these earliest vascular plants, which are very small, but it's present in all modern day vascular plants, or at least it was ancestrally present if it's in all the modern vascular plants. And that structural strength contributes to the ability of sporophytes of vascular plants to get large and to be extensively branched. And so they can become more dominant organisms in terms of they can occupy more space, they can gather more resources, both above ground in terms of harvesting light, getting more leaves out into the light, and they can also get their roots, well, they actually evolved roots too. There aren't any roots in the bryophytes. Roots are an innovation that happened also subsequent to the evolution of the earliest vascular plants like this, which managed to acquire their nutrients, we think, by an association with mycorrhizal fungi. So roots also evolved in vascular plants, and the lignified conducting tissue that's also present in roots is also important there. All right, so all these features together be pretty important, especially if you occur in a drier environment than the kinds of environments that land plants might have experienced early in their evolution. And I'll talk about in a moment how we actually have evidence that the environment did become drier about the time that the vascular plants really took off evolutionarily. All right, so let's look at a couple of major clades of vascular plants to start off with. And these are, the vascular plants are basically everything above or to the right of this node 2 here. So all of these groups. The bryophytes, remember, is this paraphyletic group of liverworts, hornworts, and mosses. And all of these plants are the vascular plants. And everything here is land plants. So here are the seed plants we'll talk about shortly. But there are two lineages that branched off before the evolution of the seed. And these are the lycophytes, which used to be called the fern allies. You look at old textbooks, or you might have even heard them called that in high school biology if you talked about study plants there. They're now called the lycophytes because they're really not allied with ferns at all. The ferns are here along with a couple of groups that we now realize are ferns too, called the horsetails and whisk ferns. But as you can see from the phylogeny here, the pterophytes, the fern clade, is more closely related to the seed plants than it is to the lycophytes. So these free-sporing vascular plants that don't have seeds, that disperse by spores, and that have to have freestanding water in order for fertilization to occur because they have free-swimming sperm. These organisms basically represent two different clades of a paraphyletic group here. Okay, so that's sort of the general outline of the phylogeny of vascular plants here. And this is a molecularly based tree. That's how we know the lycophytes are sister to all the others here. But morphologically, there's long been the suspicion that the leaves of ferns share features with the leaves of seed plants that indicate a more recent common ancestry of the ferns to the seed plants than any of these groups have to the lycophytes. We're not going to really get into that, except that the leaves appear to have had a very different origin from fusion of different branches in the case of these groups here. All right, so the seedless vascular plants then come in these two flavors, the lycophytes over here and the ferns and relatives here, these two clades. And the lycophytes, as you can see over here, look very moss-like. There is one group called the quillworts. It looks more like an onion, and it grows in shallow waters. Even here in California, I'm not showing you a photo of that. Most of the species look like this. And they've been called club mosses and spike mosses historically because of that similarity to mosses in appearance. But remember, the green photosynthetic, so-called leafy-looking parts of mosses are the gametophyte generation, whereas what you see here that looks moss-like is a sporophyte. So this is a diploid as opposed to a haploid organism. So this is a superficial similarity in appearance. And these are pretty small organisms also, like mosses, even though they're sporophytes. They only get to be, well, most of them are only, say, a foot high or so. They don't get to be too large. They have extensive rhizomes typically, but they can occur in a wide diversity of habitats. You can find them from the high Arctic all the way to deserts and up into high montane regions. So they do occupy a wide diversity of habitats. You might wonder how they can survive in the desert. And in fact, some of these are capable of drying out just like the moss gametophytes can and become essentially completely desiccated and then resume photosynthesis shortly after water becomes available. They can rehydrate incredibly quickly. Some of them are called resurrection plants for that reason. So they do have the same ability to withstand extreme desiccation in some cases, although that's not true in many. And this group was once much more diverse than it is today. As you might guess, as an early branching lineage, there are only about 1,200 species today. But as I'll show you in a moment, there were a lot more back in the Paleozoic, and they were some of the most dominant organisms in the vegetation back then. Now the ferns and relatives are about 10 times as diverse as the lycophytes in terms of recognized species. There are about 12,000 rather than 1,200 species recognized of the ferns. And they span a really wide array of morphologies, as you can see here. There are these weird things called horsetails or scouring rushes that are native here in California. They're easy to see, and that have these stems that are hollow, and they branch. They have a whorled branch pattern that makes the stem look like a tail of a horse, for example. That's why they're called horsetails. There's silica that impregnates the cells and makes them really tough and resistant to damage. And they were they're commonly called scouring rushes too, because early pioneers used them as scouring pads to clean dishes, et cetera, because they are so tough. But you can see that they have their sporangia in these terminal cones, which are pretty distinctive. But there are only about 15 species of these things. Again, a group that was once much more diverse, as we'll see shortly. This thing here, the whisk fern, used to be thought to be a living fossil that was very closely related to those rhinophytes I showed you, the little matchstick-like things that didn't have leaves or roots. But it turns out that, to the great disappointment of a lot of botanists, this thing is actually just a really modified fern. And the similarity of those fossil extinct organisms, these earliest vascular plants, was just superficial. But most ferns look something like this. And superficially, they might look like they have above-ground stems with a lot of leaves. But in fact, most ferns, everything above ground level is just the leaf. It's an individual leaf coming out of the ground, and then the leaf is just highly dissected. The blade of the leaf is highly dissected, and the stems, except for one major group called the tree ferns, which are tropical, are strictly have strictly underground stems or rhizomes. And ferns also occur in a really wide diversity of habitats, but like the lycophytes, they have to have freestanding water for sexual reproduction. So they tend to be in moister environments than the seed plants in general. But there also are, like in this group, there are some desert ferns, and some really interesting ones, for example, out in the California desert. And one of the ways the ferns have managed to cope with desert environments is, again, with this desiccation tolerance. Even here in the hills right behind campus, we have a lot of desiccation tolerant ferns in midsummer. You can go out and see their fronds just crumpled up, completely dried up, and you might think they're dead, but they come right back to life or come right back to activity when they rehydrate. And some of these ferns have strictly asexual reproduction, so they don't have a need for freestanding water because they basically bypass sexual reproduction, which is not a great strategy in the long term evolutionarily, but it works at least for a while. All right, so now let's look at the life cycle of ferns. This is really about, well, I'm going to mention lycophytes one more time, but we don't have a whole lot more to say about lycophytes. Ferns are really important organisms worldwide, and their life cycle is a nice introduction to the kind of alternation of generations that we get in vascular plants in general. So again, the sporophyte generation is dominant, and you're familiar with organisms that look like this, no doubt, ferns in general, but you're probably not so familiar with organisms that look like that. And actually, what's shown here is roughly life size. What's shown here in life is only a few millimeters in length. So these are not microscopic gametophytes up here, so everything above the middle of the slide here is haploid. This is the gametophyte generation here at maturity. This is the diploid or sporophyte generation here at maturity. These are really tiny organisms, the gametophytes. But they are free-living, and they are typically photosynthetic. You see it's green here. Both generations are photosynthetic. There are some ferns with gametophytes that live underground and are just strictly dependent on fungi to survive, but typically they're above ground. And we'll start off here. You have the spores produced again by meiosis. And germinating, we have a lot of mitotic divisions. Ultimately, we have a mature gametophyte. And typically, these things lay flat on the ground. And they're anchored to the ground by rhizoids, which again are like in mosses and bryophytes, are not really conducting, don't have much of a conducting function. They're not roots, they're extensions of cells or filamentous cells. And it basically just attach the organism to its substrate. And the organism, just like in a moss gametophyte or a bryophyte gametophyte in general, can uptake water from basically all parts of the gametophyte. So it doesn't really have to conduct water. They're quite small, and they do so pretty well all around. They don't have a thick cuticle. And in most cases, but not all, the fern gametophytes are bisexual. So they both produce sperm and egg on the same individual gametophyte. So they have both types of gametangia on the gametophyte. They have both antheridia, which remembers the male gametangium that produces sperm, and they have archegonia, which is the female gametangium that produces eggs, an individual egg in each archegonium. And these archegonia and antheridia look pretty similar to the ones in bryophytes, just a sterile jacket of cells inside of which we have the cells that will become the gametes. And so the antheridium eventually breaks open, releasing the sperm. This happens during wet conditions, as in the case of bryophytes, when water is available. And the sperm will then swim to the open neck of the archegonium and swim down the neck canal to the egg. And the distances we're talking about for sperm to swim in bryophytes and in ferns and lycophytes is really only just a few centimeters at most, so really short distances. This is not a stage in the life cycle where there's going to be much in the way of dispersal or gene flow across distance. But then we have fertilization here with the fusion of the sperm and the egg. We'll have a zygote, and then that zygote will have a number of mitotic divisions, and we'll have an embryo state inside the archegonium. And just as in the bryophytes, the mosses, liverworts, and hornworts, in the ferns and the lycophytes, the young sporophyte that develops from this embryo is parasitic on the gametophyte, but it's only during the very initial stages of establishment in the case of ferns and lycophytes, in the case of the vascular plants. And you can see the roots starting to form from the sporophyte here, so it's getting established, and the gametophyte then dies, and the sporophyte goes on to be a free-living organism that's pretty extensive. And you can see here the horizontal stem, which by definition is a rhizome if it's underground, and roots being produced from the rhizome. And if we look at the underside of the leaflets of this blade, these dissected blades, we see these little circular regions. I mean, the shape of these varies a lot depending on the fern species, but typically on the underside of the leaves of ferns, we have the production of sporangia, and these sporangia are in clusters called sori, these little circular clusters that you see here. And here's an individual sporangium, and this one is already dehisced, so it's already released its spores. And in a lot of ferns, like the one shown here, there's an explosive dehiscence of the spores. The spores are released under dry conditions, as in the bryophytes, and this thing will go off like a catapult. It opens up under negative water tension, flings back, and then releases, and it throws the spores away from the parent plant. So they have a pretty nice mechanism for dispersal of the spores. And here you can see those sori. Individually, we call them a sorus. Each one of these little circular areas along here, here's one magnified. This is a sorus. And you can see that the sorus is made up of lots of sporangia that are poking out from underneath this shield-like structure that protects them during the early development called an indusium. Indusia are not always present in ferns, but they often are, and they're really taxonomically important in terms of their shape and